Good morning, friends. Good morning, students. In today's class, we'll discuss DNA repair, right? So there are four main categories are there. First is base excision repair. Second is nucleotide excision repair, and third is mismatch repair, and the fourth is double strand break repair, right? In all these repairs, there are four things common that will I discuss. First is you have to recognize the wrong base, right? And then you have to repair it, and then you use DNA polymerase 3 to synthesize that base, and finally comes the role of DNA ligase that will ligate that strand. So these four repairs, you have these four steps common. First you have to recognize the wrong base, then you have to repair it, and then you synthesize the base where you have removed it with DNA polymerase 3, and then the strands are ligated by DNA ligase. Now we'll discuss them individually. Now in this first DNA repair, that is base excision repair, right? In this case, a single base is damaged, right? So it can happen due to deamination, depurination, or alkylating agent, right? So cytosine can be replaced with uracil, adenine can be replaced with hypoxanthine, or guanine can be replaced with xanthine, right? So let's discuss this example practically. Cytosine with uracil. So this is our normal DNA. A will combine with T, G, C, C, A, G, and T and E, right? Or in case of defective DNA, this is one defective DNA. The C is replaced with U, right? And this is wrong base pair, right? So then comes the role of uracil DNA glycosylase, right? It will act here, and the wrong base will be removed. So there here it is changed, right? Then comes the role of endonuclease in the third step. The endonuclease will remove, and there here gap is created, the red one, right? Then in the next step, DNA polymerase 3 will synthesize this gap, and DNA ligase will further seal that to form the original, that is C with G, and that is our first, that is base excision repair, right? Now the second repair is nucleotide excision repair, right? So it can happen with UV rays, ionizing radiation, environmental factors that cause damage, and it is specifically selected for when there is a large scale defective DNA, right? So how to repair is done? First there is identification, right? So these black ones, this one, this one, this one are the defective DNA, right? So second step is unwinding, right? Unwinding is done by helicase, right? The DNA helicase with unwind the two strands. So this unwinding is there, and then comes the role of exonuclease. The exonuclease is cut from the both sides, from this side and from this side, right? So this strand is being removed and further degraded. So this is defected piece is removed and degraded. Then comes the role of DNA polymerase 3. The DNA polymerase 3 will synthesize the new DNA that is given as red, right? And finally the DNA ligase will seal it, and the DNA repair is done in nucleotide excision repair. Now we'll start with the next one. The second repair mechanism, that is nucleotide excision repair. The disease associated is xeroderma pigmentosa, right? And it is an autosomal recessive disease, and the patients are photosensitive, right? And the patient is very susceptible to the skin cancer, and this is due to defective nucleotide excision repair, right? Now we will start with our third repair mechanism, that is mismatch repair. Normally there A will combine with T by double bond, right? But in this case we have cytosine, right? So it corrects a single mismatch base pair. So C to A instead of T to A, right? So how will you recognize the base? First we'll have template strand that is methylated. So this is 3' to 5' side, and the template strand is methylated, and a newly synthesized strand is not methylated, right? It is not methylated. So this is newly synthesized strand, and it doesn't have extra methyl groups, right? Then comes the role of GATC, endonuclease. So it will recognize their wrong base. So by its exonuclease activity, it will remove from here. And when this gap is created, it is further synthesized by DNA polymerase 3, so it helps in synthesis. So 5 prime to 3 prime side, it is synthesis going. And when this strand is synthesized, and it will be further ligated by DNA ligase. So that finishes with our mismatch repair, right? The major disease that comes here is HNPCC, hereditary non-polyposis colon cancer. The defect, when the DNA repair is defective mismatch repair, the disease will come, that is hereditary non-polyposis colon cancer, right? So that finishes with our mismatch repair. Now we'll start with the fourth one. Now the last is double strand break repair, and it is very dangerous, first thing, and it results in genetic recombination as well, and that can lead to chromosomal translocation, and it can lead to chromosomal breakage and cell death as well, right? And it is repaired by homologous recombination and non-homologous end joining in double strand break repair. But homologous recombination occurs in yeast, and non-homologous end joining occurs in mammals, right? So that finishes with our class on DNA repair. So please like it and subscribe it and share with your friends for the upcoming videos, and do press the bell button below. Thanks a lot.